Welcome to the channel again. Time for disclaimer. Let's continue the gene series, and the topic is transcription. Every cell during its growth and division needs to form a lot of proteins. Information to form cellular proteins are protected inside DNA and are conserved for the generations to come. This information from DNA is copied to be taken out of the nucleus in the form of ribonucleic acid, the RNA, and this process is called transcription. The process of transcription or RNA formation involves three basic steps. Step 1, where RNA begins to form getting copied from DNA, called initiation. Step 2, where RNA continues to elongate getting copied from DNA, called elongation. And step 3, where RNA completes copying DNA and stops, called termination. Additionally, in eukaryotes, when newly formed RNA comes out of the nucleus, it undergoes changes before moving to ribosome for protein synthesis. This final additional step is called post-transcriptional modifications. Let's see some basics before discussion on the steps of transcription. The DNA, which provides information for protein synthesis, has two strands. Coding strand contains sequences that codes for amino acid addition during protein synthesis. This coding strand, also called the sense strand. Non-coding strand contains complementary sequences for coding strand and thereby serves as template from which coding sequences can be copied complementarily. Coding strand runs in 5' to 3' direction. Non-coding strand runs in 3' to 5' direction. As non-coding strand acts as template, newly formed RNA is from 5' to 3' direction, and so the direction of transcription is always 5' to 3' direction. Krabs class seems interested. Let's see why. The enzyme which does this wonder of stealing the information from highly protected DNA in the nucleus is called RNA polymerase, RNAP. It has a Krabs class structure, and with its open class, RNA polymerase has two alpha subunits, one beta subunit, one beta prime subunit, along with sigma factor. Let's see the steps starting with the initiation. In the template DNA, base sequence designated as +1 is the start site of transcription. Prior to the +1 start site of transcription, there are two specific sequences at locations of -10 BP and -35 BP, called consensus sequences that comprise promoter region of DNA. The first consensus sequence of promoter region at -10 BP location has TATAAT sequence, and the other consensus sequence at -35 BP location has TTGACA sequence. These consensus sequences at promoter region are called Pribnow box -10 BP and -35 sequence. Eukaryotes carry consensus sequences at -25 BP and -70 BP called Goldberg-Hogness box and Cat box. These promoter regions on DNA at -10 BP and -35 BP are identified and bound by the sigma factor of RNA polymerase. The binding of sigma factor to promoter regions of DNA initiates transcription. This starts melting of DNA that unwinds the strands and causes local strand separation upstream. Strand separation raises the local nucleoplasm like a bubble. This is called transcription bubble. Next step is elongation. Once transcription bubble is initiated at +1 start site of transcription, RNA polymerase continues to sequentially add nucleotides complementary to the DNA sequence, and these newly formed RNA strand elongates. This sequential addition is such that C on DNA strand specifies G on RNA, G on DNA strand specifies C on RNA, A on DNA strand specifies U on RNA, and finally T on DNA specifies A on RNA. and the elongation continues uninterrupted upstream ahead. The final step is termination. Termination signals the end of transcription process where RNA synthesis stops. It can be of two types. One involves a protein factor called row-dependent termination. The other type is spontaneous called the row-independent termination. Let's see the first type, row-dependent termination. In row-dependent termination, RNA polymerase adds a C-rich tail. Row factor identifies C-rich RNA tail, binds it and unzips it. Unzipping of DNA-RNA hybrid helix is done through helicase activity of row factor. This is row-dependent termination. Now let's see row-independent termination. In the absence of row factor, RNA polymerase continues to add GC sequences that makes the stem. This is followed by addition of a tail rich in U sequence. GC rich stem is such that one half of stem is complementary to the other half of the stem. This is inverted repetitive pattern. The complementary sequences in GC rich stem turns around to bind and forms hairpin loop structure. Strongly bonded GC rich stem region of RNA heavies down from weakly bonded U rich tail, while the U rich tail remains attached lightly to the DNA. This breaks weaker AAU bonds between DNA RNA hybrid. RNA terminates itself spontaneously and comes out. While coming out of the nucleus, primary transcript RNA undergoes changes to become mature RNA. These are called post-transcriptional modifications, and it happens elaborately in eukaryotes. Once inside the nucleus, after RNA has formed, RNA is capped at its head end with 7-methyl guanosine being attached to 5' end. This is the first post-transcriptional modification, the 5' capping. RNA is attached with the tail of adenine nucleotides to its 3' end. This is the second post-transcriptional modification, 3' tailing. The primary transcript RNA formed is called heteronuclear RNA because it contains introns that do not code for proteins and the exons that codes for proteins. The intervening segments introns are removed and the coding sequences exons are joined together. This process of removing introns and joining exons is splicing, and it is done by spliceosome. This is the third post-transcriptional modification, splicing. That concludes transcription. Knowledge is truth. Let you become truthful all life. And signing off for now.